so in terms of proficiency testing, what you mentioned a lot of the aspects. I guess this is for Alejandro. I guess first.
Okay.
So you were working with a number of groups, and so how can like the proficiency testing framework be best developed from your experience, like expanding on what you said from before?
Like, you mean how to make the samples?
Yeah, like how
Proficiency testing.
Well,
for the proficiency testing. Yeah, let's go with the samples and addressing, like, creating those samples. Like, for example, we're in discussions with, like, NIST, for example, and PT Canada to try and figure out, like, what kind of standard matrix we could potentially use. But that's as far as we've gotten. We know that they operate in things like, oh, if you want to get, like, oil, you know, like, we've got a standard for that, you know, kind of thing. But how are you guys handling that, like, in Australia?
sure. Okay, so I guess there's very well-defined guidelines on how to do proficiency. Like that?
Yeah, awesome.
So there's well-defined guidelines on how you would do a proficiency testing scheme under the ISO 17043, and they outline how you should communicate to the facilities that are going to be involved, how you do the blind, how do you do the blind samples for them, and how do you share those samples with them to ensure that chain of custody is maintained and that the quality of the scheme is reliable, essentially. Essentially, what we basically did is we gave them a call, told them, would you like to be involved in the proficiency testing scheme? You're going to get this many samples, you're going to be completely blind. You need to use this SOP, and you're allowed to optimize that SOP within the context of your facility. And then it's up to you to provide me with the end results at the end. Whatever you provide with me, I'll take that as, well, gospel, essentially. That's exactly what you got. And that was across all the facilities. In terms of how you make the samples, there's two well-accepted methods. You either collect real environmental samples that could either be from an environment or from a mesocosm study, or you use, like, synthetic samples throughout. If you're collecting environmental samples from an environment, you need to ensure that the target that you're looking for in those samples is not present in that environment. You need to spike that sample later on with genomic DNA or with a synthetic oligo. And if you're collecting the samples from where, like, if your target is carp and you collect the samples from where carp live, it's background noise. So you need to collect the samples from somewhere else that has no carp, and then you can spike your sample with them. The other way is making samples from scratch altogether, in which most of the applications that I read use just simply DNAs and RNAs like pure water, and they spike them with a synthetic oligo. But I don't like that approach. Like, I feel that those matrices should have certain concentrations of inhibiting matrices, like urea. Protein, you could use milk, but milk is a problem because it causes flocculation. So DNA has a tendency to actually bind onto the milk protein. That's actually a pretty good curveball if you want to, like, really assess if a lab is capturing that. You could use salts and, like, different concentrations of salts. Is that in any way helpful?
Yeah, what we're leaning towards. It's interesting to hear your perspective on that, like, in terms of sort of the intermediate, you know, defined salts and defined urea, defined milk, tarnishing, or whatever, you know. It's a little bit different than like what we were thinking was more like along the lines of, okay, like, what standard materials are currently available for other methods that are provided by NIST and other places. Like, they have, you know, standard soils for doing certain things, and they have, like, standard water and that kind of thing. Can we convince them to make standard eDNA sample? That remains to be seen.
Yeah.
you know? So it's like actually going for the complex matrix, but it's like more standardized in that way.
Yeah. I mean, if you think about what is currently available from ISO IEC, right? There's one standard for biomonitoring that is essentially, like they send you a little vial with larval stages of insects that you would commonly find in the water column, right? In a given ecosystem. And the whole purpose of the scheme is, are you able to ID them? And if you're able to ID them, then you're proficient in the scheme. If ISO were to make one for eDNA, in whichever method, it kind of just puts this like pressure from the top, because then with that standard, then you can develop a specific proficiency testing schemes for eDNA, and then from that, countries can go and provide that service if it's something that you wanted to do. So if ISO were to create one themselves based on whatever other standards they have, that would be great. And I think that's something that Florian is actually working on at the moment, and probably people from the US as well. I don't know, quite frankly, where we're at in that space.
Yeah, I mean, that's like,
right.
We're working together with colleagues in the States to figure this out. It's really challenging. Florian, do you have any comments about that? What you guys are doing with respect to like standardizing matrices, or have you thought about that at this point?
Sure.
Yeah, we thought quite a bit about it, and there's this metaphor. I'm lacking the correct terminology. It's like the good is the enemy, the worst enemy of the perfect, or what was it? So I think we're also trying to find a perfect solution for it now, in particular when it comes now to complex community. The question is, how can we design like a reference to spiking, etc.? And I think many people want to have something that works 100%, and we will not find a 100% solution. And I think that's also a response to the question before, and I think fit for purpose means agreeing on a certain level of uncertainty. And that's what many of those manuals, maybe OECD or OIE, etc. So I think we have to agree on what's the level of tolerance of uncertainty we can accept. And then, if we agree on that, we can think about what kind of reference material would work. And to be honest, my, at the moment, pragmatic solution for, I'm speaking now of meta-mark-modeling analysis, I would actually be very happy to go for synthetic solutions because you can control them. Or maybe if it's about, let's say, phytobenthos, we can use living algal cultures, make particle counters to produce mock communities. It may not be perfect, but it will allow us to assess the variation when people analyze this very clean synthetic sample. It would be a major step forward. Thinking about this entomology paper that I just presented, where there's zero control.
Yeah, those are good points. Yeah.
We didn't know what we were doing when we started doing a bunch of qPCR, and so we took all of our old DNA extracts that we didn't need to voucher, and we pooled together a couple thousand of them for a terrestrial meta-environment, and then pooled another couple thousand together for a terrestrial, or for an aquatic meta-environment, thinking that any, tidewater goby that's in there is really negligible once you pool 1,000 extracts together of a rare thing. And so for rare species, we feel pretty good about those. And I know that Zymo makes a good microbiome one, like a microbiome community. But like we have like 50 mils of this stuff, and we just give it out to people that we're helping to get their assays up and running. And just thinking about like, can you make something so complex that it doesn't matter?
That's an interesting concept.
I'll hold it.
Yeah, one thought that comes to mind is like in terms of like the concept of creating like a mock community or a mixture, artificial mixture. Is that where does the DNA come from? Like, does it come from G blocks? Does it come from organisms that have been vouchered? How old does the DNA? Is it allowed to be before it's too old? I'm curious. Like those all have bearing on you know, like how this is implemented and how reproducible it may be, how broadly applicable it could be. Any thoughts from the panel on that?
So I guess I'll just step on this one. So the product cannot be old. This is part of one of the rules of proficiency testing scheme providers, that those standards need to be made custom for the scheme. So we cannot use one sample that is like a year old or 2 years old. It has to be made for that. So either make it beforehand, or you order your synthetic oligos and you mix them just like a few months beforehand, then you provide that to the stakeholder. Because the thing that happens is that facilities can challenge your decision. And in challenging that decision, it's the whole topic of being held accountable. Like, how do you decide that that was positive or negative, and how do you decide that I was wrong and you weren't, right? And these things happen. So you need to have fresh, standardized samples that you're going to be using for your scheme. So to begin with.
And just to the broader audience, I'm kind of curious, like in terms of like sort of where people are leaning towards with respect to the ultimate standard in your head, in your mind. Is it going to be like a G-blocks mix, or is it going to be a mix of something more real? Like, you know, from some mixture of voucher specimens or that kind of thing.
We tried this with G-block, and if it's pre-extraction matrix, it doesn't work. G-block goes right through your filters, so you can't use it as a spike-in control pre-extraction. So I'm not sure how a mock community of synthetics, that would be the most obvious way to go, but I'm not sure how you would actually use that.
Well, actually, that brings up another point, which is, at what point do you make the community, like whatever the mock community is?
So I was going to say, that is a very key, important point of defining the purpose of the proficiency testing scheme, wherein you need to decide where in the workflow you're going to be starting to test competency, right? So the conversation we've been having so far is on a spiked sample that is then delivered for testing to the stakeholder, to the facility, sorry. If you were to test also the extraction capacity of that facility as part of the workflow, yes, synthetic oligos won't work. Or for that matter, assays that are, like you need to think a little bit more about how you would do this. And the other thing is that depending on the purpose of the scheme, you don't want to be constrictive in regards to what method is being used, because you're at the end of the day testing competency of the facility to deliver reliable results. Some facilities may use, I don't know, certain filters like made out of nylon, and the other ones may use cellulose acetate. Like, you don't want to like go down the path of trying to control for that, but rather the capacity of that facility to tell you the right answer, no matter of what it is that they're using. So thinking about the workflow is key in that regard.
The other thing that we tested that's worked pretty good, some years ago we were working with FDA on seafood authentication, and so they developed their own reference library of seafood tissues. And then the test community was really frustrated because they wouldn't distribute them, saying, well, we only have a few of these fish, we need them internally, so they have a standard that no one else can comply with. So I was trying to figure out how we could make an end run around that, and we used whole genome amplification to make an essentially inexhaustible amount of genomic DNA from a single voucher specimen. So I think there are ways that we can make very consistent DNA to put into a testing schema like this. We were just looking at the barcode region, but after multiple rounds of whole genome amplification, we weren't finding any mutations that changed the sequence at all. Now, if you're looking at the genome skimming approach, way down the road, maybe whole genome amplification would introduce some error, but for these kinds of things, I think just taking whole genome amplified, it's essentially synthetic DNA but made off of a natural template.
That's cool.
Or you get fish and make a
giga preps, yes.
Go ahead, Florian.
Yeah, or just going back to good old cloning and use maxi preps, giga preps, where you can generate grams of DNA actually, you know, from plasmids, E. coli infection. So that would be a small difference, of course, but it would work. Yeah.
Yeah, there's a lot, and I was facetiously suggesting that we could use fish shakes, and just kind of make a slurry and then filter that and distribute the filters, kind of thing. So yeah, I mean, this is one area that is quite important to make sure that, yeah, oh yeah.
I've never really done a lot of fieldwork for eDNA, but if we're thinking about traditional ecological assessments, even though a lot of the time it might seem standardized, that exactly how it's being done in the field may not be. So I'm wondering if there's any way we can take field sample for traditional assessments, how they develop their protocol eventually, and if there's anything we can take away from that, because all the field biases, observation errors, they're the same, doesn't matter what kind of sampling you're doing. So I'm wondering if there's any kind of connection, or how do they standardize their unit, or something from that sort of things.
So in my case, one of the ways that we have in our facility to address those issues is using endogenous controls at the point of sample collection. So, like, you're right, like there's going to always be something that goes on when you're collecting the sample. And no matter what, that will always be the case, and you have to be ready for that. So that's why controls in the case of eDNA are so key in this aspect. We use an endogenous control. It's fairly cheap. Like, you just buy yeast from the supermarket, extract yeast DNA, and then we spike that yeast DNA into an endogenous control in the field, and that goes through the whole process. So we take that from sample collection to extraction to amplification and so on, and if at the end I'm getting let's say the copy number that I should be expecting that I should be getting, then everything went well. If I don't, well, I screwed up somewhere. And it's more of a matter of, well, how do you understand at what point you made that mistake that I feel matters in terms of what you take from the normal traditional assessments? How do you write the incorrect, visual observation, or do you write up, like, I don't know, all the many things that may happen? But that's one of the methods that we have. Did that answer your question, or did I just completely go in a different tangent altogether? I went in a tangent, didn't I? Yeah, sorry.
Do you have anything you want to add?
Well, maybe since we went on a potential tangent, would you repeat the question?
Field ecology, it was field ecology sampling methods versus eDNA field sampling methods.
I'm not technical, so that's not me.
So, I mean, I could say some words on that, because in Germany we actually use now the traditional biomonitoring experts to do the sampling, and it really depends on what you want to sample. So for biofilm, for example, it's amazingly compatible. So the way they sample, they just have to use fresh toothbrushes more often in order to collect the material, because otherwise the DNA of the old samples will still be on there. They typically do not filter water, so that's something totally new. But for the rest, also for, let's say, invertebrate, insect, or soil sampling, it's just that you need to adjust the methods a little so that the preservation is better. But we found that often it's quite easy to tweak the traditional field protocols to a way that you can work with DNA or the eDNA.
Dealing with soil is really fun when you're trying to think of field sampling for eDNA, because everyone has a different idea of what sampling soil is, even if you show them videos and protocols and you take them out and you're with them in the field. If I say duff, and then sample 10 centimeters deep, that to a fungal person is organic versus mineral, and they don't care about depth so much. Or if I'm dealing with a wildfire ecologist, it's, what is below the grassroot layer is the layer that matters, and people will make their own decisions ultimately. So as long as, my advice is always just as long as you record it, all the different ways that you can in the field, you use that as confounding variables for when you're doing your analysis. And so the standards there is on what the metadata are around the collection rather than trying to control the individual, because you can't.
I have a question. It's more about it's the standards, obviously. But you know, say that the Australian standard gets in the regulation in Australia, and the Canadian standards in regulation in Canada, and European the same. Like, what happens if we're trying to obtain global standardization, and if these are in regulatory frameworks? Like, I could see issues where, things aren't recognized across standards. Like, I know the CSA diving standard that happens in New Zealand. You know, so how can we address that so it doesn't happen the same way? And also thinking about countries, you know, in the global south or with less resources where these stringent standards may not be reasonable, you know? Like, how do we stop that inaccessibility? On the scale of, like, you know, the climate crisis and need for large scale biodiversity data.
That one I can tackle. So there are different levels of standardization. There's the national accredited standards, there's the international ISO standards. They all come with different levels of input. So a Canadian standard would have the regional Canadian context, the regulatory environments, the working environments. A German one would be slightly different, Australia would be slightly different. And that's where an ISO standard would be more generic versus detailed for specific working environments. So many national countries take an international standard, adopt it nationally, but then edit it to put that national lens on it. So generating an ISO standard takes 5 years. Generating a national standard takes 18 months. So sometimes an ISO standard would also take a national standard, adopt that into the working document, but then modify it for that international context. So it's very, very difficult to generate a technical, detailed standard for an international audience because of all the different working environments and regulatory environments. So you have to be a little more vague and a little more broad. But you can always tailor it specifically nationally to your working environment.
Yeah, sure, but then how, like, you know, like, what about the data sharing? You know, like, if there's a stop, you know, Canada's like, no, I'm not recognizing that. How do you address that?
Again, that just comes down to the working environments. Certain countries will not share their data, others will be more than happy to give you everything. It's just the international working environment that you just have to navigate. There's really no easy way around it.
Well, there's no real way around it, really, when it comes internationally. That's just the weight that you're going to have to thread at the end of the day.
We all do things too differently to really fully mesh. But there is alignment that can occur.
Yeah.
I just wanted to jump back briefly to Sharon's question. So in terms of adopting or taking advantage of field protocols now, field sampling, the third standard that Ken presented at the end of his talk, in terms of trying to develop, CSA Group is looking to develop standards for field sampling and drawing across different agencies, user groups, etc. There is a lot to tap into for that. So what he's trying to get going, once he can scare up enough funding, would tap into exactly what you were suggesting. And also there are other field techniques that are out there, for instance, water quality standards and sampling for environmental chemistry. There's many of those same considerations, and particularly you really have to challenge yourself against potential contamination. So there's quite a few field protocols out there that are fairly readily adaptable for eDNA questions.
Thanks, Chris.
I think we're going to have to guide this up pretty quick, or it's not going to be fair to the subsequent panel.
Yep.
So maybe one or two more questions.
Yeah, the last question, right here.
Thank you. I have a question regarding the ranking of the PT test. So, in respect to ranking, are you going to use consensus or false and true based on the reference data that you have from your own lab? Because some of in our lab we use the consensus for animal parentage testing, for example. There is no reference data. The PT organizer goes and rank based on the consensus. So there is no true and false, but if the majority of the lab saying that this type of fish is present, we go with that. So it's not true false, it's based on the consensus way of ranking labs based on their performance. So have you made a decision how you are going to rank the labs that are participating?
We have. So there's 4 things that we look for from inside the PT. Reproducibility, accuracy, performance, and then at the end is the decision, right? So the first 3 are within the data, and we analyze it in a way that if you would take one of the labs as the standard, right, in this case would be the one who makes the PT, so to speak, then from that you're going to be analyzing variation. So compared to this lab, how far away in the, let's say, CT values, for instance, how far off are the other facilities compared to this one? What happens if you change this one as the standard and you put this one on? So that's how you assess reproducibility, and you determine the error rate percentage that you would have from that decision making. The other one is in terms of what the decision making is being done. Even if you have that variation, is the lab able to reach the right conclusion? The right conclusion in this case being the decisions that the standard lab made. And in this case, that's why it's also very important to understand fully what blind samples you're going to be providing. There's an entity that has to know there's someone that has to know what those samples are. But that individual needs to remain separate from the whole PT altogether. That individual is the one that understands if sample 1 is a positive, if sample 2 is a negative, etc., right? One of the things that we've introduced for the purpose of our PTs is that we require sequencing confirmation at the end of it. And this came in for some debates. This is still debatable, but some of the facilities were getting positive detections outside the limits of detection of an assay, but they sequenced them and they came back 100% the target. So there's this conversation around, do we still accept limits of detection, limits of quantification within the context of eDNA when these 2 definitions came from veterinary sciences, and how you would optimize your assay within the context of veterinary sciences? Maybe for eDNA it's not the case, dealing how many traces you're dealing with and inhibition and so on. So it's not a consensus exactly. If all the labs say that this is this, but the individual that made the assay said, no, that's a positive, and you all said that was negative, we're all wrong. There's always someone that is going to be right in this regard, and is that individual. Does that answer your question? Okay.
Well, we could keep talking more and more and more, but I think probably people need to have a little break. So I'd like to thank you very much, panelists, for your and we'll reconvene here at 3:30.
3:30. What was that?
Thank you, Ken.
Thank you. That was awesome.